Wisconsin fast plants are small, fast-growing plants that are distantly related to mustard. Uh, Dr. Emeritus Paul H. Williams bred these for research purposes, and they are now used all over the country for various types of experiments, from simple observation to genetic research. Our experiment was for comparing the literature values provided by the Wisconsin Fast Plant website to the observations that we made of the life cycle points for the plants that we grew. We placed 3 seeds in each cell of a 4-cell styrofoam quad and placed it on a water reservoir and left it there to essentially grow. And after a few days, when we first saw emergence, we pulled all but one of each of the all but the tallest of each of the plant of quads. We made sure to record the number of leaves, the number of buds, flowers, and seed pods, as well as the height of the 4 remaining plants regularly. The goal was generally around once a day or every 1 or 2 days. After the full 40 days, which is the full life cycle of the plants as predicted by the literature values, we pulled out the plants, we collected the seed pods, let them dry, and we removed the seeds from them. After that, we placed the seeds in a dampened petri dish and allowed them to germinate there. We then counted up the germinated cells or germinated seeds in order to see how viable the plant was. Which the literature values for emergence were 3 days, and that's how long it took ours. For true leaves to start forming, it was about 5 to 8, and we were off by 2 days. Ours began on the 10th day. Buds were supposed to start beginning at 9, and ours was at 14. And then the bloom was supposed to be somewhere between 14 and 17 days. Ours bloomed on the 21st, and then we also discovered that one of our 4 plants had died, so for the rest of the calculations we could only use 3. The seed pods were supposed to start developing on the 18th to the 20th day, which ours was on the 24th, and from there until the end, the seed pods would continue to start growing and continue to develop, in which the 4th day is when they would be viable. Considering that's when our experiment stopped, we assumed that's when it was as well. After we did germinate everything, we had 41 total seeds, but only 18 were viable, which is less than 0.5% viability. So the plants showed a delay in comparison to the literature values, as well as an unexpectedly low viability. One of the major factors we thought played into this was unable to control the temperature, because we had left them in the hallway, which was not temperature regulated as much as a classroom would be.